Hello and welcome to the Vodcast about interphase. First of all, we're going to tackle the cell cycle. The cell cycle is the life cycle of the cell. 80% of the cell cycle is interphase. Then the other 20% is cell division. Somatic cells use mitosis, and the gonad germ layer cells use meiosis to create gametes. Alright, interphase, the 80% of a cell's life. The DNA is unwound and therefore invisible. The DNA is not in chromosomes as such because it is unwound. It's only when it becomes wound up that it forms the chromosomes. It has 3 stages. G1 is the first growth phase. The S stage of interphase is where DNA replication occurs. And then G2 is the second growth phase. After interphase, we then have cell division, depending upon what cell type we're talking about. There is some particular terminology we need to know for interphase. There is an enzyme called DNA helicase that breaks the hydrogen bonds between the 2 complementary strands. The place where it's breaking the bonds is called the replication fork. The template strand runs from 3' to 5'. The coding strand is complementary to the template strand and runs from 5' to 3'. As I said, the replication fork is where the DNA helicase is working. DNA polymerase is the enzyme that makes complementary DNA strands. There are 2 types of DNA polymerase 3 and DNA polymerase 1, but we'll get to that later. Both of these enzymes only work from the 3' to 5' direction, making complementary DNA that runs from 5' to 3'. There is an enzyme called RNA primase that is a class of DNA primases, and this enzyme makes RNA primers for the coding strand during DNA replication, which are the start of the Okazaki fragments. An RNA primer is a short section of RNA used for copying the coding strand. A short section of nucleotides is often called an oligonucleotide. Okazaki fragments are short strands of DNA created by the DNA polymerase, and they are complementary to the coding strand. DNA ligase is an enzyme that is able to join the Okazaki fragments together by creating phosphodiester bonds in the backbone of the DNA. The word semi-conservative means that each new chromatid is composed of half old DNA, which is called parental DNA, and half newly synthesized DNA. Now we're going to look at the events of DNA replication. There are a few important enzymes. There is topoisomerase that unwinds the DNA and makes sure it doesn't get tangled and broken. That works ahead of the DNA helicase. The next big enzyme of note is DNA helicase. It separates the template and coding strands by breaking the hydrogen bonds between the complementary base pairs. The site where it's working is called the replication fork. The 2 strands of DNA, being the template and the coding strand, are handled differently. The template strand runs from 3' to 5'. DNA polymerase 3 works in the 3' to 5' direction and makes a complementary DNA strand called the leading strand, aligning the nucleotides in the 5' to 3' direction. The coding strand is complementary to the template strand. It runs 5' to 3'. DNA polymerase 3 can only work in the 3' to 5' direction. Therefore, at the replication fork, whenever the 3' end becomes available, RNA primase inserts an RNA primer at short intervals. The RNA primers give the DNA polymerase a starting point. DNA polymerase 3 makes the Okazaki fragments. Then later on, DNA polymerase 1 comes along and removes the RNA primers, replacing them with DNA. Then DNA ligase comes along to join the Okazaki fragments together by joining the backbones with phosphodiester bonds. Thank you for listening. I hope you've enjoyed it. I hope it's been a help to you. And if you want to like and subscribe, please feel free to do so. Goodbye.